My name is Patrik Flammer. I'm an editor for Genome Medicine and BMC Medical Genomics, and I'm joined here by Jessica Downs from the Institute of Cancer Research in London. Yeah, thank you for taking the time.
My pleasure.
I've seen your talk earlier, and it's quite interesting to see what you're doing. Do you want to do just a brief recap of what things you're interested in?
Sure, yeah. I mean, what my lab's been interested in for quite a long time is how chromatin and the chromatin environment influence genome stability. And a major focus has been this SWI/SNF family of chromatin remodeling enzymes. Aside from wanting to understand how they function to maintain genome stability, it's really notable that they are strikingly misregulated in cancer. Yeah. So, just a couple of numbers, if you actually look at loss of function or deleterious mutations, roughly 20% of human cancers have a mutation in at least one subunit of SWI/SNF. But if you add on copy number variation or amplifications, it jumps up to 30%. So a really significant proportion of cancers have misregulation of SWI/SNF. And so we think it's, of course, important to understand what they do normally, but also important to understand what's going wrong during the process of tumorigenesis.
Oh, that's very interesting. Are there any particular cancers? Are they located in particular areas, or?
Yeah, no, that's a good question. There are, so this is a very, the SWI/SNF family of chromatin remodelers actually encompasses a few different subfamilies, and each of these has multiple subunits. Yeah. So if you look at all of these different genes, some of these subunits do have very strong associations with particular cancer types. Okay. So for instance, we've been interested in PBRM1, which is a subunit of the PBAF SWI/SNF complex, and that actually is strikingly mutated in clear cell renal cell carcinoma. So about 40% of patients have no PBRM1 expression. But other subunits are more sort of broadly spread across different tissue types. And this, again, is an outstanding question. Why do we see this pattern? Why some subunits are more prevalently mutated than others, and why some are more associated with certain cancer types than others? So it's something that obviously needs a lot more work.
Yeah, okay, that's very interesting. Yeah, because you would assume that chromatin it's in every cell. It's everywhere. Yeah. So you wouldn't expect, I don't know, would you expect a bias like that, or? Yeah,
it's a really important question, and something that, in fact, the project that we've been working on recently is sort of attempting to get at. So to some extent, it's a chromatin remodeling enzyme, so you can imagine that there are going to be tissue-specific impacts on gene expression. You know, so it might be important for regulating the transcription of a particular pathway in renal cells, but maybe not so much in, epithelial cells, or well, that's a terrible example, but, in sort of skin cells or heart cells or whatever. Yeah. So, there's an element to that. But then we also think that there are more, what we've been sort of calling core functions of SWI/SNF. And the thinking there is that some of these functions, like replication stress responses and functions, in sort of more pan-specific gene expression, and also the things that we've been talking about recently with finding a role at centromeres, those are things that you might not expect to be restricted to certain tissues or certain cell types. And so that's sort of a direction that we've been going down, and that, in fact, was the entry to the project that we were sort of currently working on, is to try to identify pathways that are more sort of pan-specific, not dependent on a particular cell type or a particular genetic context. Okay.
So if you're looking at cancers, or like the interactions in a variety of different tissues, is there any, I guess you're not specifically focusing on cancer, or it's more like the functionality of the underlying processes?
Yeah, so what our lab really specializes in, I guess, is the more sort of fundamental understanding of molecular mechanisms, and that's what we really want to get at. But I moved the lab to the Institute of Cancer Research about 5 years ago, and part of the drive to do that was, in fact, because there's such striking misregulation in cancer, we thought it would actually be really good if our lab sat next to people who are doing things that were a little bit more clinically relevant and translational. So there are clinicians and there are people doing drug discovery at the ICR. And over the last 5 years, we've developed some really fantastic collaborations with them so that we can do a little bit of both. We can actually keep working on these molecular mechanistic questions. But then additionally, when we find things that we think might be translatable, then we've got the right context to start to make that happen.
So would you also look for like discovery of these complexes in, say, like clinical cancer tissue?
Yes, exactly. So we've got collaborators who've been giving us tissue samples. So we've been doing some work with those, testing some of the predictions that we actually generate in the cell line models. To see whether or not we find the same sorts of phenotypes and impacts in clinical samples. And I'll give you an example. So one of the project that we're working on most recently, as I mentioned, is something that we found by undertaking a sort of a ground scale proteomic approach to find things that are defective across lots of different cell types and a lot of different tissue types. And we found a change in the regulation of a centromere-associated complex. So one of the things that we did to see whether or not that in fact is kind of relevant during cancer biology is to talk to our clinical colleagues about getting samples so that we could do proteomic analysis on those samples to see in cancers that are deficient for PBRM1, do we see the same sort of pattern of downregulation of these centromere proteins that we see in the cell line models that we're using. And so far it actually looks quite good.
Okay, so, that's quite promising. And I guess are you also thinking about avenues towards drug discovery or?
Absolutely, yeah. So there are some things that we can actually sort of anticipate that there will be a vulnerability based on the things that we're finding. So for instance, with the centromere defects, our anticipation is that we'll have sensitivity to the sorts of things that cause mitotic perturbations. So, you know, things that actually push cells through mitosis inappropriately, or things that actually make it more difficult for them to go through mitosis. So, spindle assembly checkpoint activities, for example. But previously, we've also found that because of the impact on replication stress responses, we tested their sensitivity to things like PARP inhibitors, and we actually find good relationships. So SWI/SNF deficient cancers show a good response, and that's been shown by other groups as well, which is quite satisfying. Again, it's sort of a more wide effect of SWI/SNF loss, so not seeming to be particular to any specific cell type or genetic context.
That's so if you see that in cancer, do you see that in do you see the dysregulation in normal cells as well occasionally, or is it one of these sort of important checkpoints for tumorigenesis, or is it
that's a great question actually. So we know, SWI/SNF complexes are pretty ubiquitously expressed in human tissues. They're required for development, so you tend to not see dysregulation during development and in normal tissues. But where people have actually done this sort of phylogenetic and evolutionary analysis of tumors and tumorigenesis, there's good evidence that at least in some cases these are very early events. And that's actually quite an important concept for a couple of different reasons. Again, one is thinking about the molecular mechanisms that drive tumorigenesis. If it's an early event, it's something that's happening that's not dependent on other cancer-associated changes. But the other thing that's quite important from that is that if we use some cell line models in the lab where we've made knockouts or mutations in SWI/SNF subunits, and we find good responses to particular therapies or drugs, then that's the sort of thing that actually might be quite clinically relevant, because you would expect all of the progeny of those early tumorigenic cells to have the SWI/SNF deficiency. So it's something actually that wouldn't just affect a subset of a cancer or metastasis, for example.
Okay. So it is quite important in the development and also in tumorigenesis.
Yeah, absolutely.
So is this? I assume then if it's quite important in development, it's quite common in not just in humans but in the wider.
Oh yeah, it's very well conserved throughout evolution. In fact, we actually did some of our early work in budding yeast on the homologs of the Swy-Snif complexes. So in budding yeast, there are two termed Swy-Snif and Risk, and some of the subunits, you can see very clear evolutionary relationship between the subunits of Risk and the subunits of PBEF. Some of the work that we did in budding yeast showing that some of these subunits had an effect on DNA damage responses in double-strand break repair, we then went into the mammalian system and actually found that those are quite conserved. So that was really satisfying, because it suggests that this is, again, these, of course there will be specialized roles, and of course in mammalian cells everything is more complicated, but these are conserved proteins, and their roles that we're looking at in terms of, maintaining genome stability seem to be quite evolutionary, conserved across quite a wide range of eukaryotes.
Okay, that's very interesting. Do they have other roles as well, or are they really restricted to the chromatin regulation?
Well, I think primarily chromatin regulation. Right. So certainly all of the work that we've done in the lab suggests that these are primarily functioning through reorganization of chromatin structure. Right. There is some data in the literature to say that they can actually act at other places on other substrates. So I think it's probably important to kind of keep an open mind about what other roles they might be doing and where and when they might be acting. But yeah, most of the things that we're looking at, we can trace very clearly back to binding and reorganizing chromatin structure at a particular place, say, for instance, in response to replication stress, or at centromeres, or at, of course, regulatory elements for gene expression. Yeah,
it's just because you mentioned that in mammalian systems everything is more complicated, and
indeed
a lot of proteins have functions that we don't really would associate with them. That's why I was quite kind of curious whether there's as it is such a key function to maintain genome integrity, they might actually have some other functions as well. No, I wouldn't be surprised at all, actually. But getting to the bottom of it is quite a challenge, because as we've been discussing, there are these multiple subfamilies. So there's, of the Swy-Snif families, there's BAF, PBAF, and NCBAF. All of them have a different subunit composition, and that in and of itself is already an oversimplification, because even within PBAF, for example, there are different isoforms of these various different subunits. So the combinatorial possibilities and then regulatory mechanisms, post-translational modifications, really mean that there are a lot of different compositions that we should be thinking about, and they play different roles at different phases of the cell cycle, and depending on location and so forth. So I think it's going to be quite a challenge, and one of the things that we're keen to do more of is so we've been using a lot of CRISPR knockouts where we just remove the entire coding sequence of one of these subunits. But one thing that we're really keen on doing more of is to actually start getting in a little bit more into the actual primary sequence to identify not just domains that are doing things, but even motifs within those domains, and sort of tiling across the proteins to identify interaction motifs, regulatory motifs, but also I think it would be quite important to get separation of function mutations, because when we do take away one of these subunits, we get these pleiotropic effects, and of course that makes it really difficult to sort of interpret or design experiments that give you very clear-cut answers about what mechanisms are at play. I mean, clear-cut results in such a system, I wouldn't expect them to be easy to get. No, it never is. We really like to have Boolean variables, and we never get them. I guess that's a research dream, to have one thing that actually gives you a yes or no answer. Absolutely. And not just like maybe or maybe not. Exactly. So your CRISPR variants, you're doing them in cell lines, I assume. So there's no in vivo.
No, we've done a little bit of in vivo work, but primarily no, we work in cell line models, and we've started to manipulate those cell lines to see if we can actually get things that are a little bit more physiologically relevant. So, doing things like 3D cell culture and using media that is a little bit more physiologically reflective of what they'd really normally be growing in, because of course the media that we normally use for cell culture is not quite.
Not quite, yeah.
Exactly so.
There's a lot of discussions I've heard about keeping cell cultures at completely non-physiological conditions in like a nutrient-rich media at 5% CO2. It's just nowhere near what's in physiological.
Exactly so. But this is actually, I think for us, it's quite an important reason then that going back and forth between interrogating clinical samples
of course,
from predictions that we would make from our cell line models is quite a useful thing for us to do, because if we make a prediction and then we can actually see it in a clinical sample, see an outcome that we expect, so sort of a dysfunction of centromere proteins or recombination events at particular sites in the genome, those things can at least give us some confidence that the cell line models are actually at least recapitulating some of the physiological functions that go on in human biology.
Yeah, I guess that's about as close as you get to sort of in vivo work.
Yes.
And I guess it's important to have these primary tumor cells as well, because sometimes the cell lines, they're not necessarily behaving exactly the same as a...
No.
But they usually don't, I guess.
Yes, exactly so. So in fact, actually, in this centromere project, one of the things that we did for kind of precisely that reason was to pick a wide array of cell line models, make PBRM1 loss of function mutations across this array, and then ask the question, which things are shared by all of these cell lines? Because of course, every cell line comes with its own issues and caveats in genetic context, and looking for things that are actually kind of globally shared gives us some sense that it's not going to be dependent on that particular context. So.
Yeah, that's very interesting. Well, thank you for taking the time and having this conversation.
Well, thank you very much. It's been a pleasure.
Thank you.